Hello everyone and welcome to a new video on the genetics of the human blood type. We're not going to look at all the blood types. We are going to focus on one of the main blood type systems. So in this video we will be focused on human blood type, namely the ABO blood type system as we learn the alleles. We'll look at how they're inherited and we'll learn some genetic crosses between these different types of blood types. So let's go ahead and get started and look at the genetics behind human blood type. So we're going to start off by looking at what is oftentimes what we can refer to as a type of inheritance called polygenetic kind of inheritances, which are multiple alleles. So we're dealing with multiple alleles. And if you don't know what an allele is an alternate form of a gene. So let's say I have a gene called blood type, and then the types being A, B, and O, these being alleles that all come together to make a blood type. Now, blood types are determined by a couple factors. So if we imagine that in blood type we have a red blood cell, RBC, and on the red blood cell there are various chemicals, some carbohydrates that are on the cell surface of a red blood cell, and these molecules are called antigens. And then what an antigen is anything that causes an immune response. Now these antigens determine the blood group. Now they're determined based on the enzyme, and this enzyme will come in and add certain carbohydrate side chains if it's one allele versus another allele. So these antigens come in A, they come in B, they come in AB. And then if they have none of them, if there is none, it will be type O. And let's look at that real quick. So let's draw 1, 2, 3, 4 different red blood cells here. And on one of them, let's draw triangles referencing our A carbohydrate. And if it's only got A carbohydrates, it is type A. If it's only got B carbohydrates, and these all being B, it is type B. If it's got B and A both on it, this is type AB. Now this happens due to the fact that our blood type alleles are referred to as being co-dominant. And we're going to talk about that more in depth, but keep that idea of co-dominant under your hats, and we'll talk about that. Now, if you have none of them, no, you were referred to as type O blood, type O. Think about that. Type O was really supposed to be type 0, but that was a typo. It has no antigens on its cell surface, no antigens. Now, these antigens, another name for these antigens that we have here, also called isoagglutinogens. Isoagglutinogens, which is why we oftentimes use I as a symbol to reference these particular things. And also these enzyme can be thought of as I. So it depends on how you want to look at that as how this name came about. Now, to do this, let's first start off and say now we have either A, B, or A and B. If you don't have any of them, you're type O. So there's really only 3 alleles. Now, when it comes to that, you have A, you have B, and you have O is the technical differences, and then you get this AB. But what is interesting about that is this, that when I have dominance, that means that a particular phenotype is produced by both homozygotes and heterozygotes. Now, I'll be doing some videos and some more genetics, but normally what we would look at is to say that I can have type A blood by being like this, being that, or being this. So I, or the isoagglutinogen, has two copies here of the A allele, referred to as a homozygote. This one only has one copy, where this I can be considered the O allele, where big I comes in A and B flavors, okay? A or B. And this would equal the blood type of A, either or. Now, if someone has IB, big B, or big I, big B, little I, they would have type B blood. So there's two ways. Both A and B are equally dominant, so they're said to be co-dominant. So to get type AB blood, you are this. That means you have two different alleles, and this is the AB blood, and this is referred to being a heterozygote. So all these in this row here are heterozygotes. That means they get two different alleles. So heterozygote, 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 homozygote, homozygote. To get O, you have to just be this. Two little i's would be the O blood type. So these are the alleles, the basic genetics behind it that says what you have. So the allele IA, big IA, codes for the A carbohydrate. Big I, big B codes for the B carbohydrate, while little I codes for no carbohydrate. And this means there are two ways to get type A blood. There are two ways to get type B blood. Only one way to get AB and only one way to get O. And that A and B are both dominant to O, making O recessive. So that's going to help us understand the genetics behind this. So let's say that in a genetic cross, what you're dealing with is, let's say we have a parent who is type A, and the other parent being type B, and they're going to have a baby. Well, here's some issues. This parent who is type A could be this. Or they could be that. And the type B parent could be this, or they could be that, to get the blood type. So here we want to do some genetic analysis. And let's say that for intents and purposes, both these parents are homozygous for A and homozygous for B. So let's say we are crossing this cross together. So we are going to cross this. Now, the first thing you want to do when you're doing a cross is figure out the product of meiosis. Now, this being one gene, one or a single cross, this has two options for making gametes. It will produce gametes only with that or that in it. So, sperm and egg would be carrying that. Same goes for here. There's only one kind of gamete that a homozygote can make. So what we do with that then is we take these and we put them into a Punnett Square, which is a probability matrix for figuring out what happens when two events are combined. Let's do the IA here, where one gamete goes at one spot and another gamete goes at the other spot. Let's just do IB, IB here. It doesn't matter which one goes on the top, which one goes at the bottom. Now what we're going to do is take this and combine it. So we would get IA, IB, and every one of these would be the same, right? All IA, IB, so 100% of their children would have 100% would have AB blood if they're homozygous dominant. Now, if we happen to have one of those options where we have I cross with IB, then we would have two different alleles and two different gametes, and we would have all the same here. And when we made our Punnett Square, we will get different results. So on that, let's just put A on the top here. Let's put the person with the B blood down here. And let's look at what would happen. Now I'm going to put it like that. And they would have AB blood here. So this person would be AB. Now this person here, we would have this cross with that. We're going to have IB little I. This person would have type B blood. This person here would be the same AB blood, where this person here would be the same as below and have B blood. So you would see that 50% of your kids are AB and the other half would be type B. So it would be a 50% chance we'd have kids with B blood with this particular mating. But let's do another one. Let's see what would happen here if we had this particular mating where one person has that cross with somebody who is this, and they make alleles, gametes, have these alleles in it, and now we will end up with very interesting results in a Punnett Square. Our Punnett Square that we do now would be quite a bit different. We would have this, and we would end up with somebody with AB blood here. We would have somebody with B blood here, somebody with A blood here, and somebody with type O. So we would have A, B, and AB, where each one has a 25% chance of occurring. Now, we're not going to do one for every single particular thing, but you have to ascribe the genotype to do the crosses. So what you would notice is, as we've broken this down, what have we seen? We saw the blood type, we saw the alleles, how they are inherited through what is referred to as a multiple allele process, and we saw some genetics of it. Now, the goal is with my video here is not to do every single cross known to man, but is to really give you the ability to do any cross that you come about. Now, would you have to be able to do this in a biology course? Yes. Would you need to be able to do this in some other courses? I know that students would have to do this potentially on entrance exams for their nursing school entrance or medical school entrance. So it is a thing you would need to be able to do and be able to understand how it comes about. Now, if you find this a very interesting topic, I do recommend that you watch my video on blood type and blood compatibilities, which I'll link in on the end of the video. But I really hope you enjoy this video and find it useful. Please let me know other topics you would like to see. Please don't forget to comment what you would like to see in the future. Let me know if you think I'm doing a good job, what I could do better on, what I could always improve, what you would like to see. Love to hear from you. And don't forget to subscribe and hit that bell button to let me know, be made aware of all my new videos coming out. I thank you so much for watching. Have a great day, and I look forward to seeing you in the next video.